All right, welcome to the second half of chapter 5 in our textbook. In this video, we're going to be talking about You Must Knows number 2 and number 4 for chapter 5. So for membranes, structure, and function, we'll talk about the role of specifically proteins in membranes. And we'll talk about the last one, how electrochemical gradients and proton gradients are formed and function in cells. So we're talking about how stuff can get across the membranes or even out of membranes in cells. So let's review our first type of transport of molecules across a membrane. And that first type is called passive transport. There's two main types, passive transport and active transport. Passive transport means molecules can get across the membrane without using any energy or ATP. Active transport, we'll talk about later, is the movement of molecules in cells using energy or ATP. Now, for passive transport, we're talking about simple diffusion. Remember, diffusion is the movement of molecules or particles down their concentration gradient. So from a high concentration to a low concentration across a membrane. So if we look at this example here, we see outside the cell, there's a higher concentration of lipid, and the lipid can diffuse across the membrane passively without any energy because it's following its concentration gradient. It's going from a high to a low concentration. Now, not every molecule can passively diffuse across the membrane, right? Because the membrane has this hydrophobic region. So hydrophilic molecules are going to have a hard time. Like glucose is a hydrophilic molecule. So glucose isn't easily going to get from outside the cell into the cell. We'll talk later about how that happens. But we know that fats and nonpolar hydrophobic molecules, they can pass through this hydrophobic region, so they can diffuse across the membrane without any energy. Also, oxygen gas and carbon dioxide gas can diffuse across the membrane as well because they're really small molecules. So that's the first type of passive transport. The second type of passive transport is facilitated diffusion, and this typically is for hydrophilic molecules. So if we have here a hydrophilic molecule that needs to get across the membrane from outside to inside, let's say this is a sugar molecule, it's hydrophilic. Well, hydrophilic molecules can't easily pass through this hydrophobic region of the lipid bilayer. And so it will use a protein channel or a channel protein to cross. And this is still simple facilitated diffusion. It's called facilitated because something's helping the molecule diffuse. It's not just happening simply on its own. So the protein is facilitating the diffusion, is helping. But it's still movement of molecules from a high concentration to a low concentration. So molecules are moving down their concentration gradient. So you see here, high to low. And we call this passive transport. No energy is needed, but we call it facilitated because the protein is helping this to happen. Now, some channel proteins only open in the presence of a stimulus or a signal. What does this mean? Well, sometimes these proteins here actually have, like, they're closed on top. They have kind of like a door. And they're only going to open if another molecule binds to this protein and opens the door or opens the gate and allows that molecule that needs to get in to get in. So think of this as like a key needs to unlock the gate in order for something to get in. So that stimulus or that signal is a molecule that acts like a key opening up the gate. The stimulus is usually different than the transported molecule. So let's say a large molecule like this shape needs to get in, a different molecule will unlock the door. These are called ion-gated channels. So an ion is a charged particle. It could be a cation, so positively charged like sodium plus, or an anion, like a negatively charged ion. And in order for those to get into the cell, another molecule will need to bind on first and open the gate. So for example, in neurons or nerve cells, there are special molecules called neurotransmitters. And this isn't super important, just kind of an example. So you don't really need to write this down, but just to have in your head, a neurotransmitter would have to bind here and act like a key, and it would open the gate to allow sodium to cross through, this ion to cross through the channel and get in. So that's called gated or ion channels. Basically, still passive transport, no energy is required, but you need something to unlock the door first. All right, now the next type of passive transport is another type of facilitated diffusion, but this is with the use of something called a carrier protein. Notice that we're still moving down a concentration gradient, so we have a high concentration of this molecule and a low concentration on the inside. So this might be a sugar molecule. No energy is needed, and so it will still diffuse down its concentration gradient, but it's going to be facilitated. And notice here, For carrier proteins, what happens is the molecule is actually going to enter into the protein, and the protein has a special shape. And when the molecule enters in, it is the right molecule. And so the protein will actually change shape and allow it to get into the cell. So this kind of acts as like a guard to make sure the right molecule fits in the right spot, like a lock and a key, and it opens up the gate to let it in. So not just anything can get in through the door, only the specific molecule can get in. Again, passive transport, no energy is needed, still down in concentration gradient, just a carrier protein opens up the gate. All right, so the next major category of transport is called active transport. Now, what's the difference? Remember, active transport uses energy or requires ATP in order to move molecules across a membrane. Now, why would we use energy? Why would we waste energy moving molecules across a membrane? What's the purpose? And what kind of molecules move across using active transport? Well, sometimes cells need to move molecules against their concentration gradient. Now, with passive transport, it's easy. If you have a high concentration of, here we have Na+, which is sodium ion, outside the cell, and it can diffuse across the membrane into the cell along its concentration gradient. But what happens if you actually wanted to reverse the process? What if you wanted to take these sodium ions and pump them out from a low concentration to a high concentration? How could you do that? Well, you need something called a protein pump. A protein pump will pump ions or molecules against their concentration gradient. So in this case, we'll take a look at an example in a moment. We're going to take these yellow sodium ions inside the cell and we're going to try to pump them out against its concentration gradient. It's not supposed to move from a low concentration to a high concentration. So this is not diffusion. This is active transport. We're going to use this pump to take these sodium ions out. In the same way, we see here that there's a high concentration of K+. This is potassium ions inside the cell. And in animal cells, this actually needs to be the case. Animal cells need to have a higher concentration of potassium in the cell. Now, if they were to diffuse out, they would move from high to low concentration. And so we'd lose a bunch of potassium ions. We don't want that to happen. So we need to actively pump these potassium ions into the cell so that we can keep up the higher concentration of potassium inside. Okay, so let's take a look at an example of how this works. Okay, so here again is our sodium-potassium pump, and the molecules look a little bit different. They're different shapes here, so let me kind of explain again. Remember, we need to have a higher level of potassium inside the cell and a lower amount or a lower concentration of sodium inside the cell. In the same way, we need a higher concentration of sodium outside the cell and a lower concentration of potassium outside the cell. So, how do we make sure that the sodium level is low inside the cell? Well, we actually have to actively pump sodium out. You have to actively pump sodium out because there's a higher concentration of sodium on the outside of the cell. Sodium won't passively diffuse from a low concentration to a high concentration, right? And so we have to actively pump it out. So how does this happen? Well, it uses the help of this protein pump here. So the first step here is we have sodium ion right here. It's going to bind to the sodium-potassium pump. And notice here that the sodium-potassium pump has a certain shape. On this side, it has a shape that allows the sodium ion to have a high affinity. Affinity means it's going to want to bind here because it's the right shape. Okay. Now, once 3 sodium ions bind, it's going to stimulate something called phosphorylation by a kinase using ATP. What does that mean? Remember how we talked about how active transport requires the use of ATP? ATP is a molecule. It's actually a nucleic acid, just like DNA and RNA. And just to remind you, the structure of ATP, we have here our sugar and our base and our phosphate groups. And remember, ATP is adenosine triphosphate because there are 3 phosphate groups, triphosphate. Now, in order to use ATP, the way ATP works is that this bond right here between the second and the third phosphate group is actually going to be hydrolyzed, or it's going to be chopped. It's going to be cut here. And so when you cut this bond, when you chop off this last phosphate, you actually get energy. And so here, the ATP is going to lose its phosphate group, and that phosphate is going to bind here to the protein, and that's going to cause the next step to work. So that only happens when you have 3 sodium ions in the right spot. It's going to trigger the phosphorylation. Phosphorylation means the cutting of this last phosphate group, and that phosphate group will bind to this protein, basically supplying energy to the protein. This is how ATP is used. Now, when ATP loses that last phosphate, now you only have 2 phosphates. So we call it adenosine diphosphate, or ADP, because now you have only 2 phosphate groups. All right, so once that phosphate group connects to the protein, it supplies energy and it causes the protein actually to change its shape. And again, this requires energy, it requires ATP. And that protein, notice how before it was kind of faced inwards and the sodium was bonded here. And this part, now notice how the sodium can't really fit in anymore. The protein has changed shape and it's reduced its affinity for sodium ions. What that means is the sodium isn't going to want to stay. It's going to want to leave now because the protein has changed shape. It's not a good fit anymore in here. And so the sodium ions are released. But notice when that happened, now we've allowed the protein to have on this side the shape which has a high affinity for potassium ions. So the potassium can easily fit in here. So in the sodium potassium pump, 2 potassium ions are going to bind to the protein. And what this does, once these kind of like these keys are in the right position in the lock, the phosphate group is going to actually be released from the protein. And it becomes something called inorganic phosphate. So it's kind of just hanging out on its own. And that inorganic phosphate can actually go find another ADP molecule. And if it were to bind to another ADP molecule, it can make ATP again. So you can kind of restore that molecule here. You can add that inorganic phosphate in order to make ATP once again. Okay, so once you have the 2 potassium ions have bonded to the protein and the phosphate has been released, notice it's been released, that changes the shape of the protein again. So having this phosphate group there or not changes the shape of the protein. So it will change shape. Now it's facing outward and then it will face inwards. Now when that phosphate group leaves, now the protein has been restored to its original shape and the potassium will actually leave because now the protein has kind of like this different shape and the potassium will have a lower affinity for the protein. And so you've actually restored this shape as well. And so now those potassium ions have been released and the cycle will repeat. Now you can take your sodium ions, 3 of them can enter in here. Once that happens, that triggers the phosphorylation or the cutting of the last phosphate group of ATP to make ADP. That phosphate group will bind on here. It will change the shape of this protein so that it opens up, releasing the sodium. The protein changes shape and allows potassium to bind, and now you have the change shape again because the inorganic phosphate falls off. Once the potassium has binded, these potassium ions are now going to enter into the cell, and now the cycle repeats again and again and again. All right, so our sodium-potassium pump that we just talked about is also called an ion pump. An ion, remember, is a charged particle, anion or cation, and these are pumps. And they do something called maintain membrane potential. What is membrane potential? Well, it's the voltage across a membrane. And what is voltage? Voltage is a measure of something called electric potential energy, or basically the separation of opposite charges. So typically in an animal cell, the inside of the cell is more negatively charged and the outside of the cell is more positively charged. So you have this voltage, you have this difference. And the idea here is if you were to take, in the inside of the cell, if you were to take a negatively charged particle, it actually has the potential to move to the positive side on the outside. So it could move across an electrochemical gradient. So if you had a positively charged ion, it will move down its electrochemical gradient from negative to the outside of the cell where it's positive. And now this is created by the distribution of anions and cations across the membrane. So if the cell is more negatively charged, that means there's more concentration or a higher concentration of anions, negatively charged particles inside the cell, and a higher concentration of cations, positively charged ions outside the cell. All right, so what this creates is a few things called forces. Together we call them the electrochemical gradient. And what this does is it drives the diffusion of ions. So ions can actually diffuse across a membrane. And that's because of the first thing, which is the chemical force. So the chemical force, chemical means molecule or particle. And in this case, this is having to do with the ion's concentration gradient. So ions will move down their concentration gradient from a high concentration to a low concentration. And that's a force. It's pushing the ion from a high concentration to a low concentration. And then there's this other thing called the electrical force, which is the effect of membrane potential on the ion's movement. So this will allow anions to actually move out of the cell towards the positive side outside the cell. All right, now we call something an electrogenic pump if it's a transport protein that generates a voltage across a membrane. Remember, a voltage is a separation of charge, a negative inside and a positive outside. And the sodium-potassium pump that we just talked about is the major electrogenic pump in animal cells. So you're going to find these sodium-potassium pumps in the cell membranes of animal cells. However, in plants, fungi, and bacteria, they have something called a proton pump. And this is in your You Must Knows, so make sure you have this down. An electrogenic pump that helps store energy can be used for cellular work. So basically, these proton pumps are going to create this thing called voltage, or basically a difference in charge inside versus outside. And this is actually going to generate energy that can be used for work. So let's take a look at an example. So here's this is still active transport. We're using ATP. And we can use something in plants, fungi, and bacteria called a proton pump. What is a proton pump? Well, notice here, outside the cell, we have a high concentration of hydrogen ions, or H plus ions, in the outside. And because there's such a high concentration of hydrogen ions, we have a positively charged outside, and there's less positively charged ions in the inside, or hydrogen ions. So we're actually going to be moving this hydrogen ion against its concentration gradient. So typically, particles move from high to low, but we're moving this hydrogen ion from low to high. We're going to use a proton pump using the help of ATP. So ATP is going to supply the energy basically to push the hydrogen on the opposite direction of where it wants to go. Okay, so this is called a proton pump, and it moves down proton gradient. All right, so just to recap, two main types of transport: passive transport and active transport. Now, we started off by talking about simple diffusion. This is where hydrophobic molecules very slowly can diffuse across the membrane from high to low concentration, and some really small uncharged polar molecules can diffuse. Uncharged or polar molecules, very small molecules can diffuse, like oxygen and carbon dioxide can diffuse across. Next, passive transport, still no ATP, called facilitated diffusion, using the help of a protein. And this is where you take hydrophilic substances. Hydrophilic can't usually cross past the hydrophobic membrane, so they're going to be facilitated. The protein makes this space hydrophilic that allows this nice, easy passage of these molecules. All right. Remember, we can either have channels or we can have these carrier proteins that will change the shape depending on the presence of this molecule here. The next type is active transport, and active transport moves molecules against their concentration gradient. So again, here we see that we are moving these molecules in, and we're moving these molecules out against their concentration gradient. So we're moving sodium out, we're moving potassium in, the sodium-potassium pump, with the help of energy. So to sum it up, once again, passive transport, just in words, diffusion of hydrophobic molecules, lipids from high to low concentration. Facilitated transport uses the help of proteins, so the diffusion of hydrophilic molecules through protein channels, high to low concentration. Active transport, diffusion against a concentration gradient from low to high, using a protein pump, requires energy, usually ATP. Now, the last type of transport we're going to look at kind of combines a bunch of things together, and we call this co-transport. Co-transport occurs when a transport protein can couple the downhill diffusion of solute to the uphill transport of a second solute against its electrochemical gradient. What this means is we're going to combine regular good old diffusion down a concentration gradient and the opposite. I'll show you an example in a second. Plant cells actually use this, and let me explain it first, and I'll come back to this slide. Okay, so we call this co-transport. So let's take a look down here at the bottom first. Remember we talked about these proton pumps? Well, what is really their purpose? What are they used for? Well, remember, we're taking these hydrogen ions and we're using ATP to pump these protons out of the cell, which makes this space really positive and this space less positive or negative. Now, once we have a lot of hydrogen ions out, they're actually going to be used by this thing called the hydrogen ion / sucrose co-transporter. Co means together or with. And so you're going to see that hydrogen ions and sucrose, this is a carbohydrate, is going to be able to get in. Now, notice here that the hydrogen ion can bind here to this spot. And when the hydrogen ion binds, it will actually be allowed into the cell. Now, notice here that this hydrogen ion is diffusing along its concentration gradient. Easy. It has a great time. It's like going down a slide. Woo, easy, right? It's easy to get in from high to low concentration. So it's diffusing. But it actually is taking along with it this sucrose molecule, and it's kind of providing the energy needed for the sucrose to get in, right? Because notice that the sucrose is moving against its concentration gradient. It's going from a low concentration to a high concentration. And so it's going to use the help of the diffusion of hydrogen ions down its concentration gradient to kind of supply, kind of like taking along with it. So imagine if you were going to go down like a water slide at a water park and you easily, you go down the water slide, but you're bringing your friend along with you and you're saying, hey, come on. And you're bringing them with you. Same thing here. The hydrogen ion's bringing along its friend sucrose. And this happens in plant cells. So think about when you think of plants, remember plants can actually create their own glucose. They create their own glucose and then they'll take that glucose molecule and they'll put a bunch together and they'll create larger molecules. Well, not every cell of plants actually have their own glucose, right? Only the green parts of the plants, the leaves, can make glucose. Those are the photosynthesizing cells because they have chloroplasts and they're able to do photosynthesis. We'll talk more about this later. This isn't super important. You don't need to write this down, but just so you know. But sugar is needed for other cells, like cells in the roots or cells in other places of the cell that aren't able to photosynthesize. And so sucrose will actually be transported to these other cells that are not in the leafy green areas, and they need to get in. And so in order for them to get in, they need to use this co-transporter protein. All right, so we're almost done here. Just kind of give you an idea of what this really looks like in the body. You can see here that you have different molecules can leave certain cells and go attach onto other cells. So when we talked about molecules kind of coming or going, where are they coming from? Well, they can come from cells that are like really close. This is called local signaling. We'll talk more about this later. You don't need to write this down, but just so you can kind of see it. Or even sometimes here's a neurotransmitter leaving a nerve cell and they're entering into the next nerve cell. So this is how nerves work, how your nerve cells work. They're passing molecules between each other. And then sometimes molecules can be sent from far distances. So here we have an endocrine cell. So this is a cell, it's like a hormone cell, and it produces these hormones. And these hormones are actually going to travel in your bloodstream to a target cell. So somewhere else that needs to go somewhere else. So they can travel from far, far away and find the new cell that it needs to bind with. And then again, those molecules can be entered in either through active transport, passive transport, or whatever it may be. One last thing I forgot to mention. So remember how we were talking earlier about voltage and membrane potential and electrochemical gradients? Let's apply some of the vocabulary to what we're seeing here. So what we have here is a voltage. So we have a positive charge on the outside and a negative charge on the inside. And what that creates is electric potential energy. So there's a potential for these positively charged ions to go into the cell because they're attracted to the negative inside, right? So we have here the electrical force is the effect of the membrane's potential on the ion's movement. So these ions are going to have the potential to move across because of the charge, electric potential. Okay. Now also, there's another force that's driving the movement of these hydrogen ions in, and that's the chemical force. Remember, there's a higher concentration, just purely by number, of these hydrogen ions out to the cell compared to inside. And so we have two forces driving the diffusion of hydrogen ions in, and this is a large force, right? We have the electric force of positive wanting to go towards the negative side, and the diffusion because of the chemical force, which is the concentration of hydrogen outside wanting to diffuse down its concentration gradient, or in this case, we call it the electrochemical gradient in. So hydrogen is flowing down its electrochemical gradient. Electro, because it's moving from positive to negative. Chemical, because it's moving from a high concentration to a low concentration. So just keep that in mind. Add that to your You Must Knows. And that's chapter 5. Good luck studying. You got this. You guys are amazing. Go you. I'm about to go down and watch the homecoming football game. Bye, guys.